Okay, so now we get to do some of my favorite stuff in biochemistry, which is good old quantitation. We'll learn some formulas, we'll get some numbers, and have a good old time. We're going to talk about ligand protein interactions. And first, I guess we should define a ligand. And so a ligand is a small molecule um, that binds to a protein reversibly. And so by reversible here, what we typically mean is non-covalent. So non-covalent interactions between a small molecule and a protein. And a protein typically is much bigger than the ligand. So it surrounds the ligand and kind of provides a space for the ligand. And it has one region of its shape that is dedicated to that protein, to that ligand binding. Usually, what happens is that the ligand causes a change in the shape of the protein, and the change in the shape of that protein causes something else to happen. So, again, one of the central tenets of biology is that the structure determines the function. So, anytime you change the structure of the protein, you're going to change that function either negatively or positively. So that interaction between the amount of protein that is bound to the ligand and the amount of protein that isn't bound to the ligand is very important towards the function of that particular protein. And so we're going to quantitate really the technically the amount of protein that's bound to a ligand as a function of the ligand concentration. We'll see how to do that in a little bit. And spoiler alert, it's going to go back to our good old Ks and uh, concentration products and things that we know and love from acid-base chemistry and chemical equilibrium. So again, these things never disappear. So we're going to use as an example of this hemoglobin and myoglobin. And so they both are proteins. We will see the structure of both of them. They're consistent almost entirely of alpha helices. We'll see how that works in a second. But they also bind to a non-amino acid moiety, right? And so they covalently bind to, in this particular case, what's called a heme group. And so a heme group is a porphin complex that binds to a iron residue, and then this iron is capable of binding reversibly to oxygen. Now, this heme group is irreversibly bound to a protein. So remember, a ligand is something that binds reversibly to a protein. The heme group, or a prosthetic group, as it's called, binds to the protein irreversibly. So we can define a prosthetic group as something, a non-amino acid that binds irreversibly. I don't know if I can spell this, irreversibly to the protein. So we don't have to deal with, kind of whether heme is bound to the hemoglobin or not, like we have to do whether oxygen is, because it's always going to be there if it's a functional hemoglobin or myoglobin protein. There are all sorts of prosthetic groups. We'll see some others as we get into metabolism. There's a lipolysine group in which you have a lipoic acid that's covalently bound to a lysine. So FAD is another covalently bound prosthetic group that is bound to protein. So there's any number of prosthetic groups that are around. For us, the key is that these prosthetic groups are irreversibly bound to the protein, and they are not amino acids, so they don't have amino acid function. And the reason for these prosthetic groups is to allow for chemistry that the amino acids don't allow. For instance, oxygen, none of the amino acids can bind to oxygen irreversibly, right? And the function of something like a myoglobin or a hemoglobin is to be able to bind to oxygen and then release it when it needs to be released, when the cell needs that oxygen. And so none of the amino acids can do it on its own, so it has to engage in a little bit of help, kind of like a consulting, gig. And they do that by hiring these prosthetic groups to come and actually do the work. All right, so here's a look of the heme group in its native environment, and we're going to see, this is the, come on, this is the protein, it's going to do all of its protein stuff. I should draw alpha helixes here, you know, however it looks. Here is another subunit, and we'll see that these are different uh, areas of the same protein. But when you look at the heme group, it sits right here. Now it's covalently bound to this histidine. So there's a histidine at, let's see, you see this. This is a histidine at F8. So this is the helix, alpha helix F, and then F8. And then there's a histidine that is there that's very important for the function of this particular, I think, I don't know if this is hemoglobin, myoglobin, but they're both pretty much the same. And there's another histidine that's down here. This one is at the alpha helix number letter E, and it is bound. Both of these histidines are necessary to position the heme group in such a way that it can bind to the ligand, which in this case is molecular oxygen O2. Now, we'll see these histidines are absolutely necessary for the function. If there are a mutation in the hemoglobin that disrupts this histidine, replaces this histidine with some other amino acid, either the distal histidine or the proximal histidine, then you can no longer bind oxygen and you have a non-functional protein, right? So one of the ways we know that these histidines are critical for the function of hemoglobin or myoglobin is because when they are removed, you disrupt the function of the protein and it can't do what it's supposed to do. Any other amino acid here, there's a potential that if you change it, it might not have as dramatic an effect on its function. And so that's how we as scientists get an idea of what are the critical residues or amino acids in a particular protein. All right, so actually, I am going to stop before we do the quantitation because I want to dedicate the time to that. And evidently, I am at around 8 minutes. And according to all the pedagogical analysis, students today only have about 6 minutes of attention span. So I'll stop and then we'll do the quantitation in a dedicated YouTube. Deuces.